Hello, I'm Danielle Galber and I'm a doctoral student at Aviv University. I'm working on developmental and epileptic encephalopathies, also known as DEEs. Our team is headed by Professor Karen Avram and Dr. Moran Rubinstein and includes 4 other graduate students and myself. My goal is to understand how a single base pair change in the DNA of the GRIN2D gene leads to DEEs. The GRIN2D gene codes for one of the subunits of the NMDA receptor, which is an ion channel in neurons. This change affects the neurons in the brain and results in a severe DE phenotype that is characterized by intellectual disability, hypertonia, and epilepsy. Currently, there is no therapy available that successfully treats all of these symptoms. This specific change has been found in an Israeli child and 4 other people worldwide. And we work closely with the family and the physicians who take care of the child to understand what the most critical research questions are. Our goal is to study how variants in this gene lead to DE and to find suitable therapeutic treatments to improve the quality of life of patients and their families. To reach our goals, we are working on several approaches in parallel. First, we're using computer analysis of the receptor and virtual drug screening, along with fluorescent-based high-throughput drug screening in cells. And we're looking for FDA-approved drugs that can be used to inhibit the ion channels that become overexcited due to the change in the gene. We're also trying to change the expression of one of the other subunits, which might lead to a reduction in the translation of the faulty GRIN2D. We're using CRISPR-Cas9 gene editing technique to precisely correct the DNA change in cells and in a mouse model. And in addition, we're using a mouse model to study the age of onset and range of the motor and developmental deficits to help us find effective therapeutic options. Finally, we're using electrophysiology to examine the brain activity of a GRIN2D mouse model to characterize neuronal changes. To conclude, we're employing various paths and technologies to enable us to fully examine the effect of the DNA change in GRIN2D and to discover therapies that can be transferred to the clinic. Working with a team of researchers and clinicians, I'm devoting my time and my energy to achieve our goal.